Prior to meiosis, all chromosomes are duplicated in a process similar to chromosome duplication prior to mitosis. Outside the nucleus of an animal cells are two centrosomes, each containing a pair of centrioles. The two centrosomes are produced by the duplication of a single chromosome during premeiotic interphase. In prophase 1 of meiosis, chromosomes become visible. Crossing over occurs and the nucleolus disappears. The meiotic spindle forms and the nuclear envelope disappears. In metaphase 1 of meiosis, the tetrads move to a plane called the metaphase plate, halfway between the two poles of the cell. Next, the spindle fibers attach to the centromeres of each chromosome. Both of each sister chromatid pair are turned toward the same pole. As a result, both attach to spindle fibers from the same pole. In anaphase 1 of meiosis, the two chromosomes of each tetrad separate and start moving toward the opposite poles of the cell as a result of the action of the spindle. Notice that in anaphase 1, the sister chromatids remain attached at their centromeres and move together toward the poles. In telophase 1 of meiosis, the chromosome pairs reach the poles of the cell. Nuclear envelopes form around them, and cytokinesis follows to produce two cells. Prophase 2 of meiosis begins without any further replication of the chromosomes. The nuclear envelope breaks down and the spindle apparatus forms. In metaphase 2 of meiosis, the chromosomes become arranged on the metaphase plate, much as the chromosomes do in mitosis, and are attached to the now fully formed spindle. In anaphase 2 of meiosis, the centromeres separate and the sister chromatids, now individual chromosomes, move toward the opposite poles of the cell. In telophase 2 of meiosis, a nuclear envelope forms around each set of chromosomes and cytokinesis occurs, producing 4 daughter cells, each with a haploid set of chromosomes. Before prophase, in mitosis, the cell goes through interphase, in which it basically just gets itself ready for the process. During mitosis, prophase is when the chromatin condenses into two rod-shaped structures called chromosomes, in which the chromatin becomes visible. Metaphase of mitosis is the process that separates duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. During telophase of mitosis, the sister chromatids reach opposite poles. The small nuclear vesicles in the cell begin to reform around the group of chromosomes at each end. Cytokinesis is the cytoplasmic division of a cell at the end of mitosis or meiosis, bringing about the separation into two daughter cells.